We're very excited to have you here today. We have a stellar lineup of expert speakers to give you information about the pipeline for hemophilia, the patient experience, and where we expect the science to go in the future. So without further ado, I'd like to turn it over to our first speaker.
So the American Society of Gene and Cell Therapy, or ASGCT, is the largest professional organization focused on gene and cell therapy. We have over 6,000 members with a mixture of researchers, both in industry and academia, and our society actively forms bridges and connections between the two. We are approaching our 30th birthday, only 28 yet, and at this point, and our goal is to advance knowledge, awareness, and education around the discovery and application of genetic and cellular therapies for human disease. And today we'll focus on how these genetic therapies have impacted on a very specific disease called hemophilia. Next slide, please. So as mentioned, we are the leading professional organization. We provide scientific content and educational programming on this broad and exciting field of gene and cell therapies. We're engaged in patient education, global outreach, policy and advocacy, which is in part why we are excited to be able to have this webinar today, and of course, host an annual meeting where vigorous and active scientific exchange occurs. Next slide, please. So gene and cell therapies, or cell and gene therapies, are really an entirely new treatment paradigm as a way of treating patients through which other modalities, such as small molecule drugs or biologics or other interventions, have proven to be ineffective or only partially effective. Next slide, please. So we categorize cell and gene therapies into two classes, so-called ex vivo therapies, in which the manipulation of the DNA of a cell is done outside the body. And then after the cells have been genetically manipulated outside the body, they've gone through very careful quality control for safety and efficacy. The cells are then giving back to the patient. And on the left-hand side is a listing of all of the approved cell therapies that are genetically engineered cell therapies that are approved in the US and Europe, along with their list prices. More specifically for hemophilia, we do what's called in vivo gene therapy, in which the drug is not a cell, but the drug is a particle of a re-engineered viral particle or some other particle that contains DNA or RNA and is injected directly into the patient, where the particle has the biologic property of migrating to the tissue and delivering that DNA content into the cell such that the cell now has a new function. And on the right-hand side are the lists of the approved in vivo gene therapies in the US, as well as their list prices. One of the reasons that these both ex vivo and in vivo cell and gene therapies are so transformative is they have been proven in patients, and you'll hear a testimony to this in our speaker panel today, to have the potential to be one and done therapies, such that a patient can get these therapies once and they no longer have to worry about their disease for many years, and the expectation is hopefully for the remaining lifetime. Next slide, please. So with that brief introduction, I want to turn it over to Bill Maurits, who's a patient advocate and a gene therapy recipient, who will tell his story about how receiving a gene therapy for hemophilia has really transformed his life. Bill.
Thank you, Matt. Appreciate it. So good morning, everybody. My name is Bill Maurits. I'm a 54-year-old hemophilia B patient who was born severe, less than 1%, and I live in northeastern Maryland. I'm a structural engineering designer who specializes in large-scale solar rooftop projects. I enjoy repairing and driving classic cars, playing backyard sports, and taking Taekwondo with my son, and traveling and going to concerts with my wife for 28 years. I'm going to give a little info about my pre and post gene therapy life experience. So being born in 1969, I've lived through the entire gambit and lack thereof of hemophilia care. In the early days, my hemophilia treatment was basically just stick me in ice. There was really not much else they could do. As treatments progressed to whole blood, they would literally, in the early days, have to wait for me to bleed out enough to be able to replace that lost volume. The next treatments progressed to frozen plasma, cryoprecipitate, etc. And then factor replacement products came along in the early 80s with, they were blood derived, and then early synthetic factors were in 92 for factor 8 patients and 97 for factor 9 patients. Treatment in my early days meant a 2-hour round trip to Baltimore, plus 1 to 2 hours at the hospital, which happened 3 to 4 times a week at least. My home treatment program was created when I was about 8, which finally let us travel and no longer had to be tethered to the local hospital. I got made fun of quite a bit in school because I would be the guy that would end up in a wheelchair for 1 or 2 days after a knee bleed and then was back up on my feet again. But my parents understood that being physically healthy and active was extremely important. I had many years of physical therapy as a kid in an attempt to keep my joints healthy. As my dad used to describe me as a Maryland crab when I was little. Parents carried that forward by having me take 4 years of weight training in high school as opposed to being able to have to do PT all the time. With that said, I was a pretty active kid, much to my doctor's chagrin. I wanted to do whatever all the rest of the kids were doing. So when my doctors pretty much said, "Don't do that," I said, "Watch me." I played full contact football, no pads with the kids in the neighborhood, baseball, street hockey, jumped and cracked BMX bikes, but always paid for it with extra infusions and time off from activities. Being physical, my early jobs were surprisingly physical. Worked at a lumber yard for a couple years, slung a sledgehammer on a survey crew, and worked as a mechanic. Obviously, as I got older, I saw the writing on the wall with joints not being so great, so I got a formal education and a sit-down job doing engineering. But being physically active when I was younger set me up for my moderately decent physical shape today. So before gene therapy, I was infusing with a long-acting factor product on prophylaxis regimen that was about once every 10 days. But more often than not, I would have to treat in between for breakout bleeds. My record was using right around $768,000 of medication in just 1 year. But I know other guys who have used more, including someone personally that said he used $2 million of factor products in 1 year. I initially researched gene therapy as a way to reduce the number of infusions I had to do, because no matter what any of us say about how easy it is to do, it's just unpleasant to have to infuse yourself. But my personal experience is going way past that. So I ended up receiving my gene therapy treatment on April Fool's Day of all days in 2016. And that was kind of weird, obviously. My first run was about 1 year with no factor infusions. After that, I believe my record was about 2.5 years with no factor. I'm now considered a mild hemophiliac and only need infusions when I do something stupid or injure myself or have a surgery instead of on a regular basis. Pretty much, I describe this gene therapy experience as life-changing. My joints are noted as very remarkable by my doctors, which is medical speak for they're really crappy. But having a steady level of factor in my system now means that recovery from activity for me is exponentially better. As a car guy, I go to a few all-day car shows during the year. Before gene therapy, I would infuse the night before, walk the show, typically infuse when I got home again, spend that night and the next 2 days with my ankles on ice just so I could barely limp into work on Monday morning. Now, I don't infuse the night before. I'm a little sore the night after the show, feel markedly better the next morning, and I'm pretty much good to go from there. My gene therapy treatment also allowed me to get a total knee replacement with minimal post-surgery infusions. Pre-Gene therapy, I would have had to infuse factor for weeks after my surgery. And after my total knee replacement, I only had to get 4 doses of my medication. And another little side note was after gene therapy, it also allowed me to get my black belt in Taekwondo in December of 2023. So gene therapy is by no means a cure, but it sure has felt like it to me. Obviously, any questions will be able to field at the end. And that's it for me. So I'm now going to pass it along to Dr. Lindsey George.
Okay, Bill, thanks so much for telling us about your experience. The disclosures that Bill and I know each other on a clinical basis, and it was wonderful to be with him on his journey. So my name is Lindsey George. I am a pediatric hematologist, my clinical practice and lab are based at the Children's Hospital of Philadelphia. And then I'm a member of the faculty at the Perelman School of Medicine at the University of Pennsylvania. And also a member of the board of the American Society of Gene and Cell Therapy. So what I'm charged with over the next couple of minutes, I just want to give you a brief introduction to what hemophilia is, although I think you probably got a great sense from Bill's wonderful outline. And then a broad brush overview of where we are in hemophilia gene therapy. So next slide, please. I think so. The hemophilia A and B are the most common rare congenital bleeding disorders. If you think of how your blood clots, you can it's an enzymatic cascade, but the sort of nice analogy is if you think of a row of dominoes, where each of the different proteins have to be in line for you to ultimately form a blood clot. And so if you take out any one of those proteins, then you can really disrupt how your blood clots. Deficiency of factor VIII results in hemophilia A, and deficiency of factor IX results in hemophilia B. The hemophilia A and B are X-linked disorders, so predominantly affect men, although not universally. Hemophilia A is about 5 times more common than hemophilia B. Hemophilia A occurs in roughly 1 in 5,000 live male births. And it has doesn't discriminate based on ethnicity or race, so affects all, all nationalities equally. And I think what you gathered from what Bill was able to tell us is that the major hallmark of hemophilia is bleeding into the joint space. So the medical term for this is hemarthrosis. So you bleed into the joint space. So what you see in the picture on, I guess, the left of your screen, you can see one of those knees, you sort of lose the architecture of where the patella is. So you basically get a swollen knee. That's what an acute bleed would classically look like. But a patient, of course, will tell you that this hurts a lot. And results in decreased range of motion and disrupts mobility. Over time, repeated bleeding into the joint space results in hemophilic arthropathy, which is essentially destruction of the joint space. And so if destruction of the joint space, you have overgrowth of the synovial tissue, so some of the soft tissue in your joint space. And the net result of that is you lose range of motion of your joints. And if you do that, of course, then you have atrophy or sort of loss of the muscle around the joint, and then you can have markedly disrupted mobility, which of course has major impacts. The most feared, far and away, bleeding into the joints is the most common of what we see in hemophilia patients in terms of symptoms. The most feared complication is intracranial hemorrhage. So this is bleeding into the brain. That's what this picture is on the right. This occurs, there's instances of this about 1% of patients every year. So you have a 1% risk per year for hemophilia patients. So relatively rare, but of course, over a lifetime is unfortunately not a rare complication of hemophilia. Next slide, please. So one thing I think that's important about hemophilia is and has made it particularly attractive for the development of gene-based therapies is that we have a pretty good way of predicting symptoms that patients will have by a simple blood test. So we can do a blood draw and measure your factor VIII or factor IX activity and anticipate the symptoms that patients will unfortunately have. So we think of normal factor VIII or factor IX activity as approximately between 40 and 150%. Severe hemophilia A and B is you have no detectable factor VIII or factor IX activity. And these are the patients that have frequent spontaneous as well as trauma-induced bleeding events. Patients in the moderate range have levels between 1% and 5%, and these patients can have spontaneous bleeding, but of course will bleed in response to any kind of surgery or mild trauma. And then patients in the mild range of hemophilia, so that's between 5% and 40%, will bleed in response to most classically do not have spontaneous bleeding and will bleed in response to some kind of trauma. There's probably some gradation between this range, where patients in the higher range of mild hemophilia have less, even less bleeding. But I think if you just click one more time, the current standard of care for hemophilia is either intravenous infusion of clotting factor. So you can infuse either factor 8 or factor 9, depending on which one of the proteins you're missing. And those infusions, patients have to give to themselves either a couple times a week or a couple times a month. Or in the case of hemophilia A, far and away, most of the patients in the United States with severe hemophilia are on a monoclonal antibody-based therapy that's delivered subcutaneously. So just sort of analogous to how you get some of your shots, so right under the skin, which is given a couple times a month. These therapies, as Bill mentioned, are quite expensive. On average, are around $500,000 a year for each patient. And if you click the next slide, or next, thank you. This is a bit of a complicated figure, but I think the main point I wanted to point out, which is that hemophilia has this long-standing history of adapting advances in DNA technology into improving standards of care. And of course, hemophilia gene therapy efforts are, I think, a really shining example of this. So we now have 3 approved adeno-associated or AAV vectors for hemophilia, 2 for hemophilia B, 1 for hemophilia A. And I think what's important, of course, all of us in this call are deeply indebted to developing and improving the care of hemophilia patients. What I think is also important in the even broader scope of gene-based therapies is a lot of what we've learned for the development of AAV vectors, which could be used for a variety of other disorders as well, has come out of work in hemophilia, of which this has been in clinical development for a couple decades. So if you go into the next slide. So I just wanted to outline briefly the current approach. So first, I just want to acknowledge that most of the work in hemophilia gene therapy is using AAV vectors or adeno-associated viral vectors. There are other approaches not using AAV that, just for the sake of time, I will not be talking about. This, again, is the predominant approach, which is outlined here. So the vector is given by an intravenous infusion. It's most classically given outpatient over the course of an hour, and then there's a series of monitoring that day. And the concept here is that you infuse the vector, it goes into the patient's bloodstream, and traffics sufficiently to the liver, which is the natural place that your body makes clotting factors. We target a type of liver cell called hepatocytes, which is the natural place your body makes factor 9. It's not the natural place your body makes factor 8, but we think these cells can do a reasonably good job of making factor 8 still. So the idea is that AAV vectors are a very efficient way of delivering DNA to the nucleus of a cell. And in our case, we're targeting the liver and we're targeting hepatocytes. The AAV vector will deliver either the factor 8 or factor 9 gene. One point I wanted to point out is that you can see from this schematic, the sort of circular piece of DNA is meant to symbolize either factor 8 or factor 9 DNA to encode either your factor 8 or factor 9 protein. Predominantly AAV is non-integrating, which is a positive when you think about safety considerations if DNA were to integrate into the wrong place. One of the challenges, though, is that if a tissue is rapidly dividing, say, in, for example, an infant, and this is a congenital disorder, this approach right now is really only being developed for adults. And I'm a pediatrician, so we hope in the future that we can bring a similar approach to help younger children with hemophilia. But again, the point is that you have infusion of the AAV vector into the bloodstream, you target the liver, deliver factor 8 or factor 9 to a type of liver cell called hepatocytes for expression of factor 8 or factor 9. For factor 9, just to point this out, if you start looking at some of the information for hemophilia B, we're encoding a naturally occurring variant of factor 9 for the 2 approved or FDA approved vectors called factor 9 plasma. And this is a variant of factor 9 that has improved activity. So the concept here is you get more bang for your buck, so to speak. If you could progress to the next slide, please. This is, I tried to put together some of the aggregate data that we have out of clinical trials. And I think a lot of this work tells a very similar story. So the endpoint of clinical trials for hemophilia is looking at your annualized bleeding rate, which is exactly what you might think it is. It's just looking at the number of bleeds you have every year. Both of these graphs for both hemophilia A and B show your annualized bleeding rate for a number of different AAV vectors, both before and after gene therapy. And although the numbers vary slightly, they overall tell the same story, which is that if you have sustained factor VIII or factor IX expression, you have a marked improvement for patients. And universally, if you have sustained expression of factor VIII or factor IX, you reduce and in some cases eliminate bleeding in patients. So I would say that we have had multiple proof of concept successes. And then, of course, we've now progressed beyond that to have 3 approved AAV vectors for hemophilia. So if you go to the next slide, please. And then I just wanted to show, maybe click one more time. I just wanted to outline, so both in terms of what these licensed therapies mean for ensuring patients have access to them, but also with respect to resources needed, federal resources that are needed to continue to support this work and have been really instrumental to where we are with current successes. So federal resources, both at the level of the NIH and the FDA, have been really critical to develop this work. And you might have gotten a sense of this as I was briefly going through this, but the hemophilias have really been a model single gene disorder for the development of gene-based therapies, particularly the use of adeno-associated or AAV vectors. So there are 6 approved AAV vectors. Half of those are for hemophilia. That's probably not a coincidence. But I'll just note that there are now more than 200 AAV vectors in various phases of clinical trials worldwide for more than 30 disease indications. And a lot of what we learned in hemophilia does translate to other disorders. So I think my point to you is that I think hemophilia provides a nice example of where we might anticipate AAV vectors expanding for other disease indications. And I've just listed some of the things we've learned out of hemophilia trials that apply more broadly for this class of therapeutics, things like understanding the human immune response to AAV, creating capability to manufacture these vectors, creating a regulatory framework, et cetera. So of course, we have a long way to go, but some of the successes of where we are now have translated outside of hemophilia as well. And then if you click one more time, please. I should note that this figure here is meant to outline the number of different organs that are being targeted for various diseases. So you can see this work has expanded quite a lot. And then specific to hemophilia, we have this remarkable success as I outlined in the last slide, but we anticipate that this work will continue to only get better. And so I think one of the critical points of the example of hemophilia gene therapy is, of course, now biotech and large pharmaceutical companies have invested in this work. But many or nearly all of the critical advances and some of the basic science and translational work have come out of largely NIH-funded labs. And so it's been really important to have that support to progress this work. And then lastly, this table is just outlining the number of hemophilia A and B gene therapy trials that are either recently completed or active and ongoing. And you can anticipate that this is putting a heavy demand on the FDA. And so having access to expertise to review these protocols and just the sheer volume of them has been really important. And we appreciate the sort of more recent investment in the FDA to develop capabilities to meet the demand of ongoing gene therapy work. And so hopefully some of those points might be a good transition to Len Valentino. So Dr. Valentino, I believe, is going to tell you about potentially next steps in hemophilia gene therapy.
Thank you, Dr. George. That was an excellent overview. So I'm Len Valentino. I'm also a pediatric hematologist, and I'm the former president, chief executive officer of the National Bleeding Disorders Foundation. I'm currently an entrepreneur in residence for a venture philanthropy program at the National Bleeding Disorders Foundation called Pathway to Cures. And this is an opportunity to bring innovation into the bleeding disorder space. So I want to build on what Dr. George talked about and first emphasize that there's still a number of unmet needs in hemophilia. And in particular, people who don't have access to gene therapy. So bleeding continues to be a major problem. Bleeding causes pain, as she alluded to with those wonderful pictures. And next is those bleeding into joints causes joint disease. And this is a crippling arthritis that can be devastating, reducing the quality of life for people, but also limiting their ability to participate in daily activities of living. The treatment of hemophilia, along with the disease, creates a significant burden. And I think our two patient representatives here will have, one has already shared some of that burden with you. And I know that our second patient will also be able to share some of the highlights or lowlights about the burden of disease. And then lastly, inhibitor formation, which is an immune response against infused factor, remains a major problem. So how does gene therapy address any or all of these issues? Well, it starts with a better understanding of the disease state. Dr. George began to discuss this, and this begins with innovation and research. And innovation and research really is focused in academic university research centers and laboratories, and partnership with industry. These laboratories are able to make significant advances. One thing that I'd like to note is the very important legislation, the Bayh-Dole Act, which allows nonprofit organizations such as academic medical centers to own, patent, and commercialize inventions that are created with federal dollars through that are granted from the NIH. And this is a major incentive for academic medical centers to be able to continue this type of work in innovative areas. So this partnership between academia and industry is vitally important to continue. A great example of this is the program that was started between the National Institutes of Health and the Food and Drug Administration, the Bespoke Gene Therapy Program, or the Bespoke Gene Therapy Consortium, which is an opportunity to really bring together the best minds in the field of gene therapy to create new pathways and shorten the regulatory pathway to approval for new gene therapy products. So the NIH is critically important in all of this, and continued funding of the NIH, and in particular for hemophilia, it's the National Heart, Lung, and Blood Institute that's so vitally important as our leadership in the NIH. The Food and Drug Administration is the next area that I focus on. There's currently 31 gene therapies that have been approved, in addition to 30 RNA therapies have received regulatory approval. But that's only the tip of the iceberg. At last count, there were 2,068 gene therapies in development, and that includes 66 for blood disorders. So the task for the FDA is very substantial in terms of monitoring all of these clinical trials, but then doing the regulatory due diligence for them. The next is the importance of the Centers for Disease Control and Prevention. The Centers for Disease Control and Prevention is critical for surveillance and data collection around people living with bleeding disorders. This data is collected through the Hemophilia Treatment Center network, and that data that's collected then goes in to helping inform where the gaps in care are and where innovation can occur. And academic medical centers, again, lead the way in terms of developing new information. Well, even if gene therapy was available to everybody, but it's not. So one category of individuals who don't benefit from gene therapy currently, as Dr. George alluded to, are children. And these drugs have only been approved for adults with hemophilia. You can also take the other extreme of age, where elderly patients, because of comorbidities, may not be eligible to receive a gene therapy product. There's also this pre-existing immune response to the AAV vector that may preclude certain individuals from receiving gene therapy. This is a very active area of investigation by industry partners as well as academic researchers to try to figure out why this immune response occurs, and then how to overcome it or how to deceive the immune system and allow gene therapy to proceed. Dr. George also showed you that the target cell is the hepatocyte in the liver. Well, if the hepatocyte is damaged or sick, it's not a good target because it's the factory that will make the transgene product, the gene therapy product. So we need to have a healthy liver. And we all know that there's an epidemic of fatty liver disease in the United States as well as globally. So dealing with liver disease, even before the patient considers gene therapy, is something that's vitally important. Next is the issue around inhibitors. So the gene therapy clinical trials that have been done have excluded people who've developed an inhibitor to either factor 8 or factor 9. Well, you wonder how often this happens. Well, approximately 25 to 30% of people with severe hemophilia A, that exact population who would benefit most from gene therapy, develop an inhibitor against the factor 8 and therefore have been excluded from the clinical trials. Again, this is an area that we need more innovation and investigation to figure out whether we can actually provide gene therapy as a means to induce tolerance in these individuals who have inhibitors. So a major area of investigation. Another area is 75% of people living with hemophilia globally don't have access to any care. They lack diagnosis. These are people who live in low and low middle income countries in the developing world of Africa, Southeast Asia, Latin America. Because of the lack of access, they don't have access to gene therapy either. And this may be a population that would definitely benefit from gene therapies in the future. So again, we need to research and innovate how to bring gene therapy to this very important population throughout the world. And then lastly, the cost. Dr. George had a little bit of information on her slides about the cost of these. And much of the cost is related to the development and creation of the gene therapy, running of the clinical trials, and then the manufacturing of these products once they receive regulatory approval. We need to figure out how to do this more economically from beginning stages of innovation, running clinical trials more efficiently, and then manufacturing, which is one of the major goals for the bespoke gene therapy program. How can we bring down the cost of manufacturing these products so that they can be accessible to more people worldwide? So I think those are some of the challenges where innovation and research still are needed to bring gene therapy to the entire hemophilia population, but more importantly, to people who are living with rare blood disorders throughout the world who could benefit from a gene therapy product. With that, I'll end and turn it over to our next speaker. Thank you.
So I think while we're waiting for Bobby to log back in, Caitlin, can you, can I ask all of the speakers to turn their videos on, and then we'll just Q&A until Bobby's able to log back in. I just want to make sure that we all have an opportunity to ask some of these questions. So I'm going to kick off some of the questions that we've received. Bobby gets logged back in, we can ask him to give his audio presentation. So I'm going to start with you, Dr. Porteus. You mentioned at the very beginning in vivo versus ex vivo, and I know that we've talked quite a bit about in vivo, but I'm going to, while this briefing is focused very much on hemophilia, I'd like to bump the conversation out a little bit, just broadly about cell and gene therapies and the delivery. Examples of cell and gene therapies using ex vivo and kind of where we're at with the landscape in that right.
Sure. Thank you, Margarita. So actually, it's a very, I think, important question. So there are a number of different types of genetic blood diseases. So we're talking today about hemophilia, which Dr. George and Dr. Valentino, others have explained, is a disease in which you're missing a protein in the blood, and so you don't clot, so you end up getting bleeds into your joints or tragically sometimes in your brain. But there's other genetic blood diseases that don't affect clotting, but affect how cells behave. They can affect how your immune system functions, so-called immunodeficiencies. They can affect how red blood cells function, and those are like thalassemias and particularly sickle cell disease. And so when you have a genetic disease of the blood that affects how the cells function rather than the proteins in the blood, that's when we have to use an ex vivo gene therapy, because we're going to give stem cells of the blood, manipulate them outside the body, and then give them back. So the resulting red blood cells or immune cells that derive from the stem cells are now genetically repaired, so they make normal red blood cells. So we have, even though these are two categories that one can lump as genetic diseases of the blood, one is cell-based and really needs a ex vivo cell-based therapy, and one is protein-based, and really the best way of currently treating it, although Dr. Valentino highlighted we still can do better, is by using an in vivo therapy that treats the liver in order to treat the blood. And so it's not that there is a correct or perfect one-size-fits-all, but cell and gene therapies need to be tailored to the underlying genetics and disease pathophysiology.
Great. Thank you very much, Dr. Porteus. I see Bobby has rejoined us. Bobby, we're going to pivot a little bit. And what we're going to do is ask you to give an overview of your presentation, just using maybe your cell phone. We will imagine, we did have a great picture of you on our first slide, but maybe if you can just walk us through your experience having received gene therapy for hemophilia, and we will advance the slides as you go through your presentation.
Can you hear me now?
Oh, yes, loud and clear.
Can you hear me?
Yes, you're good to go, Bobby.
Okay. So what's interesting, today's presentation is on gene therapy and personal experience. This audio kerfuffle is a prime example. I'm going off slide for a second, just bear with me. That there was a plan already in place. There were briefing meetings, things set up. Something happened that disrupted everything, both in a good and a bad way. There was an adaptation to make this work. That is gene therapy from my personal experience. Because growing up with hemophilia B from back in the day till now, that is where I functioned in. That is where, in some conversations, I was forced to look at the positive side versus the negative side. It could have been easy a couple of minutes ago to say, I'm not going to finish this call. However, because of what's been poured into me, to say, no, let me go through this process, this journey. That is gene therapy. That is the bleeding disorders community, because of what is on the other end of it. Yes, you all probably see slides, you know, lovely PowerPoint that was created. And one of them in there talks about a cast iron. Some of you who are old school or know what a cast iron skillet is, and that what it does and does not do in its capabilities. There are certain things highlighted in that slide, words for a particular reason, as it relates to my journey in gene therapy, which was not easy, but it was done because of many voices in the conversation, because of those whose shoulders I still stand on. So the question came, how is my experience, you know, unique or different? I believe Bill presented his life experience. We're both hemophilia B, yet still have different experiences. That is the essence of gene therapy. And the once and done, yes, on one end, it is a once and done. But on the other end, there's multiple once and done, both from the standpoint psychosocially, both from the standpoint educationally and work-wise. It's not an easy A plus B. There are nuances to it for each individual person. The person does not stand in this journey alone. It's both the clinical team, it's family and friends. And so the question becomes of what does gene therapy do? For me, it fosters conversation. It fosters the continued ability to look at what is possible. And it changes the dynamic. Because back in the day, I was taught to treat early, treat often. And the clinical teams I had out of UC San Francisco taught us how to be independent, regardless of our medical conditions. They taught us how to fight in many different ways that did not require physical strength, but to continue being in and of community, where your bleeding disorder did not nor does not define you. So that is what I got for now, seeing as how we had a kerfuffle.
Fantastic. Thank you, Bob. And if we can, Caitlin, if we can ask all of the speakers to turn their video cameras back on, and we'll do a Q&A. I know that we have some really great audience participation here, and we can take the slides now. Thank you. Great. So Dr. George, I'm going to pivot to you. You had a lot of really great information in your presentation, and I applaud you for trying to squeeze all of that information down into 8 minutes. I know that's not necessarily the easiest thing to do. So obviously we've heard from from Bill and from Bobby about the patient experience and how their lives have changed since receiving a gene therapy. Can you talk a little bit more about what the conditioning is required of a patient prior to receiving a gene therapy? So we know that for some other treatments, they require different conditioning. So can you talk a little bit about what leads up to a patient actually receiving the therapy?
Sure. I want to, if anyone has familiarity with some of the ex vivo approaches that Dr. Porteus was alluding to, conditioning, I would say, is more about anticipation and planning, and making sure the patient has appropriate information to make the best decision possible, but there's not any medication conditioning. So I would say that, and maybe Bill can speak certainly more to this than I can, but I would say that to me, what's most important is that the patient really understands what the commitment is, which is really, I think Bill and I had this conversation that, I was going to be virtually a bit part of his family over a year of close monitoring. And so, which was great, by the way. I think that, but I think what's important is that you make sure the patient is appropriately consented. And I think that matters whether it's a clinical trial or that's a licensed product for sure. And so basically just what does that look like? It means the patient should understand the nuts and bolts of what the therapy is, what the monitoring will be, what restrictions this will have on their current day-to-day, what we know in terms of outcomes as it relates to both safety and efficacy, what the sort of range of observations are in the patients. And then I think the other critical piece is that, the goal of this is for this to be a so-called one and done therapy. But I always sort of say this is the obligation as where we are in the current state of science. So at best we can tell, you probably cannot get this again. And, that may change certainly with scientific developments. So I do think there's a large gravity in the decision, in particular based on that. So again, there's not a medication conditioning where you decrease someone's blood cell count or something of that nature, but the conditioning is more a lot of conversation, a lot of making sure people understand what the process will be, and then also to make sure the patient has the availability to have close follow-up and monitoring.
Sure, absolutely. And the monitoring and the follow-up is a critical piece in the field. Can you share a little bit with the audience, what are some, and if somebody was interested in participating in a clinical trial, what sort of questions do those families have in terms of what helps weigh their decision of participating versus not participating?
Sure. I think, I cut out for a moment, but I think what you're asking was what is the monitoring entail? And I think, so that's the first part, I think, of what you mentioned. So in the first couple months, typically the first 3 months, the monitoring is classically blood draws once or twice a week, and just regular communication with the team, your clinical team. So I don't want to overplay the commitment, but it is a significant commitment just in terms of having blood draws and being in frequent contact with the clinical team. And then those labs space out a fair amount. And what we're monitoring for is so make sure your liver enzymes most far and away haven't been major safety concerns in terms of how your liver functions. But we're monitoring for that, but also has some implications on making sure that therapy is effective and works for you. And then I would say before you receive the therapy, there's a lot of monitoring to make sure that this is a safe and best approach for you in terms of making sure your liver is healthy, like Dr. Valentino is alluding to, or you might have other comorbidities that make us think that this is maybe not the safest route for you. And I think the second part, forgive me, what was the second part of that you asked me to explain, or let me know if I hit it already?
Actually, I think that you did hit it. I'm sorry for the technical difficulty today. Really just one of the things that are often asked are like, what are the questions that patients have when they're making a decision to get a clinical trial? So what are some of the, so you hit all of those key points in your response there. I'm going to pivot to Bill, actually, since we're talking about patients and recipients of gene therapies. Bill, thank you first and foremost for sharing your journey. I think one thing that people may be interested to know, received your first gene, since you received your gene therapy, and what has that impact been? You shared the impact to yourself, but can you talk a little bit about the impact to your family as well? Because we know that oftentimes people with chronic diseases, it's not just only the person that impacts the family.
Sorry, if you could repeat the first part of that question. You kind of lost audio for a minute. I just want to make sure I get everything that you're asking me for.
Sure. Can you share with the audience how long it's been since you received your gene therapy? And then can you also talk about the impact to your family?
Impact on the family, I guess it really wasn't a whole lot. I've always kind of been a relatively independent physical guy anyway, so I've always been one of those people that kind of never let my hemophilia slow me down. I consider myself a patient with hemophiliac, not a patient with hemophilia, not a hemophiliac. It's certainly shaped who I am today, but it doesn't define me. And I'm sorry, you still broke up on the very first part. Were you asking me about some experiences?
How long it's been since you received your therapy?
I received my infusion in April Fool's Day of 2016.
Margaret, if I might just follow up with Bill to get your input. I know you sort of described this a little bit of just your ability to do karate with your son or do some activities with your son. Do you think that changed pre and post gene therapy, or has impacted any of those activities that you all do together?
The activities themselves haven't really changed, just how I feel doing them and how I recover from them. You know, my ankles are in pretty bad shape, so I can't really run, but I still try and move around the backyard playing soccer with my son and football and everything else. So I won't say my physical ability has increased, but again, as I said previously, my recovery after, you know, I'm just not sore and I don't feel as bad after doing a bunch of physical activity. Same thing with going with doing karate. You know, one of the things we have to do is we have to spar. So we put on gear and we try and beat the snot out of each other, basically. I'm not sure I would have been able to do that as easily pre-gene therapy just because of bruising, etc. But you know, it's on the back end for me that personally has made the biggest difference.
Well, I'll open this up to Dr. Valentino, Porteus, and George. We talked at various stages about the role of translational science in cell and gene therapy, the value of the Institutes of Health. The Bespoke Gene Therapy Consortium was also touched on. Can you share with our audience like what the role of translational science is in developing these cell and gene therapies and the value that it has? And if you want to expand a little bit more about the Bespoke Gene Therapy Consortium, I'm certain people would be very happy to hear that as well.
Want to take the first part, Matt?
All right, go ahead, Len, you got it.
So, I think the collaboration and partnership between academic medical centers, and whether it's industry or the funding that comes from the NIH is critical. This research would never happen without funding that comes from the federal government. So that funding is vital to the progression of knowledge and innovation. So, it is really the fuel that, lights the rocket that leads to these discoveries. And, the initial work that was done, more than 20 years ago in gene therapy was done under the auspices of, NIH funded grants. And it's really important to recognize how important that is, not only in the past, but today, that without federal funding and federal dollars, this work would not happen. So it's, that academic work is critically important. But there's clearly a role and a place for that industry partnership, because industry is, again, if you think about the rocket analysis, that's the titanium, the booster rockets that are really going to get you into space. And they have the manpower, woman power, and they have the financial resources to bring these things to, the finish line. And that's so important. So there's usually begins in an academic setting, and then there's a translation into an industry or a commercial setting where the work continues. And I think that's been a really wonderful model to follow, where academic medicine then partners with industry, and then there's a handoff. And a lot of times it's an academician who is the founding member of a company because of the importance of that knowledge that they bring to the company. So they're oftentimes the chief science officer or sometimes the chief executive officer. I think the Bespoke Gene Therapy Program, that consortium is a wonderful opportunity to bring together people who are really vested in trying to create best practices. So the consortium's goal is really to shorten the time from sort of inception to regulatory approval, and doing things to create better pathways for manufacturing. Creating a playbook, for example, was one of the first goals of the consortium, of how this can be done, taking best practices from various sources. So the Bespoke Gene Therapy Program runs under the National Center for Advanced Technologies, NCATS, and I think that's a really important initiative that's been undertaken. There's an opportunity for continued partnership in that, and it again is a great example of academic and industry partnerships working together. There's also a large component of what are called clinical research organizations, people who are experts in running clinical trials, who participate in that, who are sharing their knowledge and experience about the best ways to get clinical trials done the quickest way possible and the most efficient way. So a lot could be said about that Bespoke Gene Therapy Program, but I think the overall goal is to get these medicines to people who need them quicker and more efficiently at a lower cost.
And if I could just add one thing to what Len said, the other, I think, really unique and important aspect of the Bespoke Gene Therapy Consortium is the active involvement of the regulators and the FDA, that they are at the table trying to come up with solutions, not just passive recipients of ideas. And I think that's really, well, I don't know if it's unique, but I think it is a powerful aspect of having all those stakeholders at the table trying to figure out problems together.
Thank you both. And I'll also say we, ASGCT, is a proud founding member of the Bespoke Gene Therapy Consortium. So So we promised to the audience, we did not plug that into Len's presentation, but we are very happy he mentioned that.
Yeah.
So with that, I know that we are at the hour. We have a lot of great resources, reference, and so we will follow up with the email to everyone who is registered. We are also going to post a recording of this on our YouTube page and recirculate as well. I know that we did receive several messages of people that were interested in attending, but this is a very busy day on Capitol Hill as well, if anybody's watched the news. So thank you all for your time. Thank you to Bobby, Bill, Matt, Len, Lindsey. Very much appreciate you sharing your experiences from all aspects of cell and gene therapy delivery and receiving it. Thank you, everyone.
Thank you.
Thank you. Thanks.